All right, hi everyone. Welcome to our workshop today with a Stanford PhD student, Mr. Njoo. He's going to be starting off by presenting a few slides about what he does. So I'll hand it off to Edward, and then we'll do a Q&A session afterwards. So if you have any questions, make sure to have those handy.
Yeah, so thank you all for having me. Can you all hear me okay? Yes? Can I get a verbal yes, just to make sure I'm not speaking to a wall? Great. Okay, so I'm going to I'll talk briefly about my background, and then this is largely for all of you to ask me what science is like. I'm actually not going to talk much about stuff from Stanford, as many of you know. So I did my undergraduate and master's in SoCal and came back up here for my PhD in Stanford. I am going to talk about some of the science that I'm working on at a place called ASDRP, which many of you might be familiar with. We're in Fremont, and I manage a very talented team of high school students here at this institution, and I'll be sharing some of the ways in which we chemists talk about and think about science and about therapies for things like cancer. So just by show of hands, how many of you are students who wish to be doctors maybe when you grow up? Some of you? Okay, 3 people want to be doctors. That's great. When I was your age, I certainly wanted to be a doctor myself. I actually, went through the whole pre-med thing and MCAT and the whole bit. And then I decided part of the way through my college experience that actually, I didn't realize, but you could be a scientist as a career, and that made all the difference to me. So I'm going to share briefly. Let me see if I can get my slides to load. Can you all see my slides?
Yeah.
Great. Okay, so how do I go to full screen? Interesting. All right, as long as you can see them, then we're okay. I do have a lot of tabs open. Maybe if I do this, it'll be better. There we go. Can you all see the slides still?
Yeah, we can see them.
Okay, great. So this is I'm going to talk briefly about how chemists how we chemists think about things like drug discovery and therapeutic science. I think when I was in high school, like many of you, I heard chemistry and I thought of this esoteric subject where people sit there and crunch stoichiometry numbers and KEQ and they calculate things that nobody else on earth cares about. But as I think the farther along you go, the more you realize that actually science at the interface of chemistry and biology and medicine is quite an exciting area to be involved in. So I'm an organic chemist by training, which means that I'm interested in molecules that have a lot of carbon-carbon and carbon-oxygen, carbon-hydrogen bonds in them. And we chemists think at different levels. There's some chemists who like to think about really small molecules. Things like Taxol, maybe some of you have seen Taxol before. It's a very potent anti-cancer drug. Doxorubicin, these are in the clinic. But these are some other compounds that we worked on in the group that I manage here at ASDRP, Menastral, and this guy's called LAS. And these are preclinical compounds that also destroy cancer cells. And then you have the world of biologics. Hopefully all of you are vaccinated. If you're vaccinated, you have a biologic inside of you, or you had one at one point in time. Things like proteins and nucleic acids, which are much larger than small molecules. Then this is just the interesting pictorial of a small molecule, aspirin, in comparison to an antibody, which is a biologic. One's a lot bigger than the other, obviously. And a lot of these small molecules that we work with as chemists actually come from nature. I like to say we've been doing drug discovery ever since cavemen have been running around in the woods looking for trees that cured people of disease. And that's actually how a lot of small molecules today in the modern 21st century make it into the clinic. In fact, Taxol, Dox, and etoposide, these are 3 very popular anti-cancer drugs that are typically a part of some of these chemotherapy regimen, actually come from plants or from fungi in nature. And in fact, this is an open access paper, by the way, that you can access online. A great deal of small molecules which are used to treat human disease today, these green segments, almost 1/4 of them are either coming from nature or they're inspired by or designed from those which are made in nature. And so I'll be talking about a few of the products that we've been working on. This is not necessarily just a plug for ASDRP. This is a plug for chemistry and for thinking broadly about science. I think when we're in school, we think that, to make an impact in human health, you have to be a clinician. And I have a great deal of respect for clinicians, but what drove me to be a scientist was realizing that in between my two gloved hands, I don't have gloves on right now, but in between my two gloved hands, I could innovate and I could design structures that would have new functions and new ways of treating human disease. So one of the small molecules from nature that we've been interested in this group is this molecule called Andrographolite. It's a small molecule NF-kappa B modulator. Many of you know that NF-kappa B is a transcription factor that is responsible for a lot of cell survival genes, genes that are responsible for telling a cell when to divide or when to stop dividing. And obviously, this is of great interest in cancer, but also in other indications, including multiple sclerosis and HIV/AIDS, for which this compound is already in clinical trials in Europe and in South America. But as chemists, we were more interested in not just the fact that this compound is a very good anti-cancer agent, but also in the question of how does this exactly work? And so as a transcription factor modulator, we've been interested in asking the question, how does this actually impact the genes that are being expressed? And so we've done this qPCR experiment where we can actually track, this is pretty cool, you can actually track the expression of mRNA as a function of time in raw cell lysate. Here's another compound. This is not an ATFA product. This is one that was actually discovered over at Harvard Medical School about 20 years ago. It's called Menastral. And you are all familiar with this sequence of cell division, prophase, metaphase, anaphase, telophase. What happens, interestingly, is if you take a cell that's happily in metaphase, you can see the chromosomes lined up across the middle, and you treat it with a micromolar concentration of this small molecule Menastral, you see complete, exiting of the cell cycle, and the cell, instead of forming these beautiful spindle fibers, gets absolutely destroyed. So that's pretty good if you want to treat cancer, because cancer is nothing more than uncontrolled cell division. Now, one of the things that we've been particularly interested in is not just making Menastral, which, by the way, we made this on gram scale. It binds to this motor protein called kinesin EG5. I think the compound costs several $100s for 10 milligrams on Aldrich. We made 5 grams of this in our laboratory just for fun on a cloudy day. But we were interested in not just making Menastral, but also analogs of the compound that might be better. And so we've been collaborating with a company in Canada called Analysis, where we developed ways to develop reactions that enable rapid construction of these scaffolds, which have very potent anti-cancer activity. And so we've been working on these two sorts of compounds, the Menastral type compounds, as well as this known anti-cancer agent called Carmophor, which is a hexyl isocyanate derivative of 5-fluorouracil. But we've been using this instrument called nuclear magnetic resonance to actually track the formation of products in this chemical reaction and identify new reactive intermediates that show up in a route in making these molecules. Okay, I'm going to talk briefly now about our biologics program and kind of some of the things that we've been working on with antibodies. mRNA vaccine, hopefully many of you are familiar with that technology, but I'm going to talk primarily today about protein therapeutics, things like antibodies, ADCs, fusion proteins, peptides, and peptide hormones that are used in the clinic. Specifically, I'll be talking about monoclonal antibodies and how we chemists have been modifying them for clinical use. So a lot of cancer drugs they have a lot of off-target toxicity. We all know this because if you have family members or friends who have suffered from cancer, one of the problems that they have is the side effects of chemotherapy causes hair loss, all these terrible indications that are actually collateral damage from the chemotherapy rather than the cancer itself. And nature actually has a pretty good way of dealing with the selectivity problem. The problem is, we've been throwing toxins at cancer cells for a long time, and these toxins kill all kinds of cells, not just cancer cells. Well, your immune system actually produces monoclonal antibodies in general that target specific antigens. And the Nobel Prize in the 80s was awarded to these group of scientists who actually developed a way to mass produce antibodies, the same weapons that your immune system produces to target kind of rogue cells. in a laboratory. And this is how we make monoclonal antibodies, antibodies with a single antigen target. You take a progenitor cell that responds to an antigen, you fuse it with a myeloma cell to make them immortalized, and then these guys become antibody mass-producing cells. Antibodies are cool, but they're kind of they're kind of boring. They just stick to stuff. And so one of the questions that chemists asked a few years ago is, can we actually take an antibody and throw a very toxic drug onto it? Okay, and why is that important? Because if you have an antibody, it's going to give you absolute selectivity for a cell that you want to target. And the drug, you can have this as toxic as you want, and as long as you can target this toxic payload just to the cancer cell that your antibody hits, guess what? You can induce cell death in that cell and that cell alone, and not all the other cells you don't want to kill. But this is a chemical problem at the end of the day, because one has to then develop chemistry that can link a very complicated anti-cancer agent with a linker that can be released at will when it hits the cell. And so this is a chemical problem at the end of the day that has largely been solved and is currently being solved by organic chemists around the world. This is fairly new technology. So ADC's antibody drug conjugate technology, it began in 2000, so just over 20 years ago with Mylotarg, which is a calicheamycin ADC that it has a disulfide linker here that's cleavable. But in the last, if you look at these dates, in the last 22 years, ADC technology is rapidly accelerating. There's only 11 approved to date. And out of those 11, half of them were engineered in the last 3 years. Isn't that crazy? This is very new and cutting edge technology. And so our lab here at ASDRP has been interested in making these antibody drug conjugates. This is one such example where we take a very toxic payload. It's a mitosis inhibitor called MMAE, monomethyl auristatin E. We link it with this linker. This is a peptide cleavable linker. And upon enzymatic cleavage, this protease cuts this amide bond. It does a 1,4-methide iminoquinone elimination, and you get the antibody and the drug. Now, the beauty of this is your payload is attached to the antibody up to the point where the antibody hits the cell. And when the antibody with the payload gets intaked into the cell, these proteases, which are found all over the cell, cuts this bond, and you release toxin and you destroy the cell you want to destroy without impacting collateral damage. So this is one of several generations of ADCs. This is actually previously published by a different group. And unfortunately, some of the technology that we've been developing, we're still pursuing patent on it, so I can't talk about it yet. It's under non-disclosure agreement. But this is stuff that I find interesting. I hope that this is stuff that I think we as chemists know that this is going to revolutionize the future of drug discovery. So other things that we, I guess some of the considerations that we chemists think about on a daily basis in terms of problems that we need to solve: molecular design. Can we design a synthetic route in a chemical entity that can be released at will? Okay, isn't that cool? You can take a molecule and get it to do what you want it to do simply by designing a chemical structure that reacts in a particular way. Scalability. We're beyond the days of making single-digit milligrams of compound. If you make a milligram of a compound, that's not going to solve any problems in medicine. But if you can make a gram of a compound, you will. Safety, step count yields ways to make molecules in more efficient manners. And we've been exploring other ways of releasing the drug as well, including using pH labile linkers, photo cages, so you can use photons to release these things, different enzymes, enzyme selectivity, etc. So that's kind of some of our ideals in chemical synthesis. So I hope in that very brief presentation, I captured some of the ways in which we chemists think about drug discovery. And this is now in collaboration with the biotech company, a lot of which I could not share because of our NDA agreement. But we as chemists are in a position that I think is very exciting, where one can design and build and test small molecules that have interesting and novel function that might either compete well with or outcompete existing therapies out there, with the ultimate goal of let's solve real world problems, which is that people are still getting cancer and still dying of cancer. Okay, so I'll take any questions that you all might have. Well, I could either call on people or they can, I guess you can feel free to shout it out. Zave has his hand up.
Hi, yeah. I actually had started a PhD program. I was supposed to do organic chemistry, like at a university that's not really well known for that. At the time, or really even now, the guy that I was supposed to work with was basically one of the only people doing any type of synthesis there. And like right before the program, something with funding or some stuff like that, I wasn't able to work with him. So, you know, I kind of cut the program, and I'm just kind of wondering, like, I guess, like, what would be your advice for someone who wants to get into primarily synthesis when, like, it seems like so many chemistry programs are kind of dominated by, you know, biochemistry and material sciences?
That's a great question. So Zaid, I assume that you're a postgraduate, right? You've finished an undergraduate degree and you're looking for things to do afterwards.
Yeah, exactly. Yeah.
That's a good place to be in. I think, you know, on the one hand, on a philosophical level, you know, doing a PhD is maybe useful in some regards, but degrees at the end of the day are simply reflections of training. And I know a lot of very talented scientists, some of whom I work with, who I have a great deal of respect for, who don't have PhDs and who I think make just as significant contributions to science. You make a very good point, which is in the modern day, it's harder and harder to fund synthesis for the sake of synthesis. And I think that's because people tend to look at organic chemists as this exotic, maybe esoteric breed of scientists that care about questions that nobody else really cares about. I think the answer to that is, you know, even biochemists and materials people, at the end of the day, if they're going to innovate, they need to make molecules. And the question is not whether or not making molecules is useful, or I think the better question is how do we engineer our synthetic efforts to make the maximum use of the compounds that we're making. So I think you make a very good point, which is that it's harder and harder to find organic chemists, but at the same time, I think chemists are being more interdisciplinary. With the example of an antibody drug conjugate, that was stuff that's developed by biochemists in conjunction with synthetic chemists, or biochemists who know how to do a little bit of synthesis. So, you know, I would encourage you to think broadly about science, and that it's not as segregated as one might think. It's not as if the organic chemists are here, the inorganic chemists are here, and the material scientists are off on a different planet. People in the modern world appreciate scientists who can gather information, gather expertise from different backgrounds, and solve problems.
Oh, thank you. Yeah.
Other questions?
I've got one. I'm from the UK. I'm in my final year of my undergrad. I'm in a sort of similar area to you. I'm making porphyrin photosynthesizers for PDT. I'm just wondering, I'm currently like writing it up. So I was just wondering, like, what's the best way to improve your academic writing? Because at the moment I'm like sort of struggling to like get up to like when I read papers and things like that, like I can't really compare mine to theirs. Like I feel like it's not as developed as it should be, if you know what I mean. What would you say the best way to do that is?
That's a great question. So, and thank you for joining us from the opposite side of the world. I didn't know that there's going to be a lot of undergrads here here. You know, I think sometimes science doesn't necessarily work the way that one wants it to work the first time. I can speak from personal experience. I think almost every single experiment that I've ever done in my undergraduate or graduate career at some point in time didn't work the way that I wanted it to work. And that's okay. I think we as scientists need to be less driven by, I guess, outcoming images of success, and more about people who can troubleshoot through the weeds. Sometimes, you know, if I meet two scientists, I enjoy talking to the people who enjoy a great deal of success in what they've already done, as well as people who have tried everything possible in the world, and yet everything seems to fail, or they feel like there's a glass, you know, ceiling on where their science can go. Because I think scientists are uniquely in a position where we're constantly learning. And every experience, whether it's one that hits the ceiling really quickly because things are just not working out, or things that fly to the moon and end up in a Nobel Prize or other great things like that, are valuable experiences for for we as humans who want to solve problems. Does that kind of make sense?
Yeah, for sure.
If you're applying for graduate school, sometimes telling the story of, gee, all the stuff that I tried, I wanted to do the following. It just, sometimes science doesn't work. But I think people who go through times of things that don't work, they realize more about the nature of science than anybody else. Yeah.
Yeah, thank you. I can relate to that quite a lot, to be fair.
That's good. That's good. Okay, other questions? Go ahead, Rashid.
Yeah, so we kind of talked about how anti-cancer therapeutics have evolved in recent years, but I want to get your take on where do you think it's going to be in the future, maybe in like the next 20 to 30 years.
Yep, so that's a good question. I think when we as a society began anti-cancer drug discovery, we started by asking the question, what things kill cancer cells? And that's not a bad question to ask, but it's a very simplistic way of looking at cancer, right? We spent decades of research developing toxins and inhibitors and things that just destroy cells. Somebody who I greatly respect once said, basically, if you throw lead nitrate onto a cell, that's a pretty good anti-cancer agent because it's going to kill a cancer cell, but it's also going to kill a lot of other things. So I think as a society, as a scientific society, we're moving past, rote things that just destroy stuff and more towards targeted therapeutics, small molecules that can either selectively kill cancer cells through modulating mechanisms of disease. Right, we're at a point in science where we don't have to throw things into a black box. We understand mechanism as chemists. Or things like antibody drug conjugates, where you can deliver something to selectively attack a tumor. And I think that's where the trajectory of science is headed. We're past the days of throwing random stuff at cells and seeing what sticks. We are in the days of being able to design rationally small molecules that can have specific functions. Yeah. So whether that's an mRNA vaccine for cancer one day, or whether that's a small molecule, or whether that's a biologic, these are all worthwhile pursuits.
Yeah, thank you. Makes sense.
Yeah, I guess we'll go Mahir and then back to Zev.
Yeah, so another question that me and Rashid had was, like, in the past years, we all know that technology has advanced tremendously and impacted like pretty much every part of our lives. But we know that scientific research especially has been impacted by this new wave of innovation. So we also noticed that on your ASDRP profile, you did a lot of work in like computational and theoretical chemistry. So do you think you could talk a little bit more about the relationship between computer science and chemistry and how you think like future advances will impact this field?
That's a fantastic question. So I think, as scientists, we need to be open-minded about getting skills that we normally don't put into categories. And one of those skills that I had the privilege of getting trained on when I was an undergraduate was in computer modeling. How long do you think it takes to make a molecule? Anybody want to guess? A minimum, maximum time?
Go on, Zev.
Oh, sorry. I was just going to say, like, actually synthesizing it, I guess, depends on the complexity, but something to get to market is years and years and millions of dollars.
Thank you. Yeah, no. And this is how drug discovery traditionally works, right? You hire a team of, scientists who sit there and they make a bazillion compounds and slam all of them at cells. And the synthesis takes a ridiculous amount of time. Every student I've ever mentored realizes that the hard way, that, making a molecule isn't the job of one afternoon usually. It's usually the job of a team of people over the course of several weeks, if not months, if not years. So developing things in a laboratory is very time consuming. It's very financially consuming. I spend a lot of my day trying to figure out how to fund projects at ASDRP. And we're thankful for a lot of companies that have donated for that. But synthesis is very time consuming and expensive. In the meantime, you can use computer simulation and get information about a chemical structure in a matter of hours. We have a computing cluster here at ASDRP that's very powerful, and that's like basically a supercomputer. But we can sit here and screen a few hundred chemical entities in about 2 days. That's a lot more efficient, not only in terms of time, but also in terms of material, personnel, than it is making a few hundred compounds. If we were to try to make those few hundred compounds, it would take us years. And so I think the role of computational modeling has really been effective in accelerating the drug discovery process, because if you can ask a computer to simulate something, you can arrive at results, information faster that will then inform what you do in the laboratory. It's a good question, though. Okay. Zave has another question.
Yeah, I was just because one of the ways you mentioned of delivering the drug, so you conjugate the drug to the antibody, and then I'm assuming once the antibody is taken into the cell, it's then enzymatically cleaved.
That's correct.
How do you deal with I guess selectivity issues when dealing with something like that? Because one of the classes I took was medicinal chemistry, and we were, with ADME, we had to think of all the different ways, like how will this drug, once it's in the system, be chemically transformed by enzymes, whether they're P450s or whatever they are. So I guess, how do you deal with, like, especially when you're talking about an extremely toxic drug anywhere else, that sort of enzyme selectivity elsewhere in the body?
That's a good question. So selectivity is a broader question beyond just enzymes, acid, labile, photocage, protecting groups. This is one of the reasons why we've been interested in photocaging these ADCs, because light, nothing in biology emits light, I guess, unless you're a firefly. And so that's one way to achieve selectivity. The reality is, once you stick a molecule onto an antibody, it's a very different chemical environment. It's kind of protected almost by the antibody. In fact, sometimes the challenge is not keeping this stable enough. Sometimes the challenge is actually getting this off, because when it's stuck to the antibody, this is not drawn to scale. But if you imagine this antibody being, 100,000 times bigger than the linker, it's kind of shielding the linker from the chemistry that's happening elsewhere. So you make up a good point, which is that maybe floating serum proteases might cleave some of this off. And that's certainly one consideration that one has to take. So typically, this is the job of a lot of analytical chemists and biochemists. They do stability experiments. They ask the question, how stable is this to different conditions? Some of the technology that we've developed here uses acid controllable release. So we can actually control the pH dependence of a release of a drug. I can't talk about that technology on the slide deck yet. But that's another way. As chemists, we can design chemical structures. Can you actually, is this the best linker to make? Can you actually change the hydrolysis kinetics, or can you achieve enzyme selectivity? Some people have put peptides in here, so you can get selectivity for certain proteases. But there's many ways of achieving that. I think the better question, maybe that's a very good question. Maybe the core of what one might ask is, what is the best mechanism to target where you can release things at will? Does that make sense?
Yeah, exactly. Yeah. The other thing I was kind of thinking of is like induce. I'm guessing this is what photocaging is you use induced fluorescence from the antibody binding or something along those lines.
It's a little bit different. You're close. We use a functionality. Imagine that this is a functionality that reacts with the photon. And when it reacts to the photon, it comes off.
Oh, okay.
That's what photocage is. So it's a molecule
cool.
masked by an entity that comes off with light.
Okay. Awesome. Thank you.
Other questions?
Yeah, what's for the sort of things you research, what sort of molecular targets do you have, or like biological targets do you try to target?
That's a good question. I can't talk about a lot of things because this is in collaboration with the biotech company now, and we're bound by a non-disclosure agreement. But there's many things you can target. A cancer cell upregulates a lot of antigens on the surface that you don't find. And so some of the things that we've looked into are mainly those antigens, but then you have to be able to make a custom antibody that targets those antigens.
Okay.
You have to do that, but those are proprietary. So.
Yeah, that's fine. Okay, no worries. Kind of cool.
Yeah, that's a good question. Rishit has another question.
Yeah, so I have a question which is really for the audience that like hasn't done anything research related before. So basically, when you're starting a new project or you're like getting into some ongoing research, what's a good way to start thinking about how to approach the problem?
So I think scientists like to pride themselves in innovation a lot, but what we sometimes forget is that everybody is sitting on the laurels of all the body of work that has been already done. No scientist is operating in a vacuum. Okay, any project anywhere, whether it's something that I'm working on or something that my colleagues are working on, started because there's a body of scientific literature of published papers and patents and protocols that have already been developed by somebody, and we're simply building off of known information. And so I think something that's a little bit underappreciated is the background reading that one typically does to understand the field. If you're going to innovate in a field, you have to know what's already been done.
Great, thank you. That makes sense.
Yeah. Mahir, go ahead.
So another question I think many, like especially high schoolers have that are applying to college or getting into that area. So do you think you could talk about the path that you took to reach where you are? Let's say like starting from high school all the way up to now, and talk about like the application process and any extracurriculars or tips you have for people applying.
Yeah, this is going to sound very countercultural, but I'm a big proponent of, I don't really think it matters where you go for your undergraduate education. I don't, I think people like to, people who, well, I think as a society, we like to brand ourselves in terms of brand names. Okay, and this is one of the reasons why I'm not talking about my Stanford stuff, because people as a society, we're drawn by brand names. And I think I'm not saying that Stanford's not a good institution, maybe it is. But what I am saying is what's more important than the brand of the institution that one goes to is what you do with the opportunities given you there. Okay, there's some of the brightest scientists I've ever met went to like Cal State Long Beach, and they're some of the brightest people who are pioneering amazing things in science. And Cal State Long Beach is not a bad institution, but it's a very unassuming institution. When I was in high school, I wanted to, maybe like many of you, I wanted to build my own path. So I applied to a bunch of colleges. I got into some pretty good ones like UCLA. And then I ended up going to a college that paid my full way through my undergrad. So that was Loyola Marymount. That's where I did my undergraduate education. And to this day, I'm very thankful that they paid for my undergraduate education because I would not have been able to afford private school, university degree. But that's where also I got involved in research. Looking back at where that was, maybe almost a decade ago, if I had not gone to a very small, unassuming institution like Loyola, I probably would never have ended up in research because this was a field that was very foreign and, to be quite honest, a little bit scary for me. And so at Loyola, it was a primarily undergraduate institution, small school, almost very little research funding, but hey, it was free. And so that's why I went there. I got to know a lot of really talented faculty members who took it upon themselves to mentor me, not only in terms of how to do science, but also how to be an independent thinker. And it's because of those people, I'll just name some names: Wesley Dowd, who I started out with doing some, fluorescent stuff with marine biology applications. And then I transitioned to work with David Moffat and Jeremy McCallum at LMU. Those are the people who, to this day, I'm tremendously grateful for because I owe my scientific career to them. So I would encourage all of you high schoolers out there, don't worry so much about where you're going to go. Go where you're going to be happy and where you will be properly mentored. Sometimes the best mentoring for undergraduates does not necessarily happen at the world's biggest, flashiest name institutions. It happens in the small colleges where undergraduates are actually the center of the attention of what people do. Yeah, I wasn't a super smart high schooler, so all of you are probably way more accomplished than I ever was. Even being on this call, I wasn't thinking about this when I was in high school. And Rishit, go ahead. I apologize. I have to go in about 7 minutes, so we'll take a few more questions maybe.
Yeah, sure. So this is actually not my question. This is from the chat. I think someone was wondering, how was the MCAT experience if you took it?
I don't remember much except that people, it's way overrated. I think in maybe this is also countercultural, but study a subject to build expertise. Don't study a subject for the exam. study a subject because you want to master. Like if you're going to take the APUSH exam in a few days, study APUSH because you want to know American history like there's no other. And if you do that, things like I'm not saying that it's trivial, but you know, I think people stress about exams too much. Yeah. Go ahead, Mihir.
Hi, I was just gonna say, this is kind of like, since you have to wrap it up soon, that's no problem. I was just gonna say, if anyone has any questions, like this is probably the last chance to ask them in the chat. But if not, if nobody has any other questions, I guess I can, oh, looks like we have a few more. Never mind.
Okay, Chris and Zave, since you two are undergraduates, feel free to pop me an email.
That's actually what I was going to ask, if you had any your email out so we could contact you outside of the call and not take up too much time. Oh, thank you. Yeah.
If you have questions about like applying for grad school, I'd be happy to for you to, you know, I'm happy to respond to those if you have questions.
Yeah, for sure. That's been a big thing I've been dealing with. So
certainly. Yeah.
Okay, mine was more, what's your advice on trying to get yourself in some papers before I've got a master's lined up like after this year. And what's your best, what's some advice on getting into some papers before I go on to do my doctorate?
That's a good question. I think you know, papers are always good to, you know, they're like your scientific they're going to build your scientific pedigree. I think I wouldn't worry too much about papers. I would worry about finding what you find exceptionally interesting. Start by presenting some work at conferences. There's the American Chemical Society conference, happens twice a year. I'm sure that Europe has the equivalent of that. But when you start working on a project, start thinking about what is the end goal going to look like. It's not a bad idea to start thinking about the end goal at the get-go, because that's how you drive every experiment so that it's towards the publication. Right? We don't want to be sitting here spending our time and effort on experiments that are not going to end up in the paper. So think about the end goal at the beginning. Like, what is this going to look like in its final form?
Okay, right. Okay, no worries. Thank you for that.
Yeah. Great. So I left my email in the chat window. Feel free to contact me if you have questions about science and whatnot. I'm happy to answer them. But thank you all for having me. It's been a pleasure. I love talking to young people about science because I think it's, you know, I'm not that much older than all of you, and it's, you know, when I was in high school, I didn't realize you could make a career out of being a scientist, and had I realized that earlier, that would have kind of made the world a difference in how I saw science and how I saw myself. So, yeah.
Yeah, thank you so much for your time. I think we all learned a lot today. It was very informational.
Okay. Yes, absolutely. And then, Chris, if you have a question, why don't you shoot me an email, and then we can take it offline.
Yeah, I haven't removed the reaction yet. Sorry.
Okay. Okay. Well, take care. Have a great rest of your weekend.
Yeah, thank you so much for attending, everybody.